Hi all, welcome to part 2 of our lecture on sequence alignment. We have already chatted about similarity versus relatedness, what the differences between those two things are, and our edit distance. I hope y'all took a bit of a breather in between that last video and before you dived into this one. I know I did. You could tell by the end of that last video, my words were getting a bit jumbled, my brain was getting a bit jumbled, but we got there in the end. So we're going to move on now, start talking about dot plots, and then we'll get into sequence alignments and end with substitution matrices. So dot plots are essentially a way for us to visualize the similarity between two sequences. It is truly just a way for us to plot on like a grid, like we did in math, our x, y coordinates or axes, and then we go through, we would have the different sequences like the A's, the T's, the G's of one sequence, and maybe we have a T, T, G of a different sequence, and we would just go through and act like a matrix here and put a dot on things that are similar. It's a little bit more complicated than that. We'll get into the details there, but that's the gist of it. So we could see here we have an A and a T, those aren't the same, so no dot. Here we have an A and a T, not the same, no dot. An A and a G, not the same, no dot. The only ones that start being the same are this T matched with this first T, this T matched with the second T, and then with this G over here, we have a G as well. So this is a very simplified, kind of messy example. I will go over other ones in the future in a few slides here if that didn't make sense. But it's effectively just kind of chunking it out like a matrix, putting one sequence on the y-axis, one sequence on the x-axis, and then adding dots wherever the sequence nucleotides are identical. So here is a much nicer overview of what I just explained. So you'll see over here on our x-axis, we have the sequence A, C, T, C, G. And then on our y-axis, we have the sequence A, C, A, G, T, A, G. And these dots represent when the nucleotides on, we'll call this one sequence 1, and this one sequence 2, match up. So anywhere there isn't a dot means that those nucleotides aren't the same. So we can see, for example, if we take this T here, and why we don't have a dot in this box here is because if we go all the way up in that column, we see that the nucleotide for that other sequence is a C. Since C and T aren't the same, we don't get a dot there. But similarly, if we look at T in this line here and where we have a dot right here, when we go all the way up to sequence 1, we see that that also has a T, which is why we get a dot there. So in these really small, simple sequences that aren't very long, it can kind of seem like this is just a bit of a mess, and I don't quite understand why we would be doing this, Prof. Kim. But it will become clear as we do some examples here. So one thing that I want you to think about and that I posted in the course discussion page is what would two 100% similar sequences look like on a dot plot? What would sequences that are similar look like on the dot plot? So if you want to prove anything to yourself, you can feel free to build out those matrices with two 100% similar sequences before you, or 100% identical sequences before you answer that question, but just try to follow your gut. What do you think it might be? It's okay to be wrong. This is probably a very new topic to everybody in this course. We're going to have some wrong intuition there, and it's good for us to kind of unpack and talk through that and figure out why that intuition is maybe misleading us in these different cases. So post in the discussion board your thoughts around those two questions. One complication when using dot plots in practice is that once we start using really large sequences or larger than what we've seen, say, on this example here, they can start getting a lot of noise. So if you just go nucleotide by nucleotide, you start seeing a lot of dots all over the place, and it can be really hard to pull out the pieces and the areas of similarity. So to overcome this, we use a sliding window and a threshold technique in bioinformatics to compare more than one nucleotide at a time. We're able to adjust the window size and the threshold to kind of tweak what kind of similarity we want to be trying to capture in that dot plot. So this is just a text overview of what that means and how we would actually put that into action. I'll read it through and then we'll get into an example after this of what this actually means. So at the beginning, we always have to define what our window size and what our threshold is. Our window size is just the number of nucleotides we're going to take into account at one time. So this instance, we're saying we're taking into account 10 nucleotides at a time. And then the threshold is how many of those nucleotides out of the 10 need to be the same in order for a dot to be present. And we're saying in this case, 8. So if we had two sequences that were very, very long, we would still build out that matrix where we have our rows and our columns and our two sequences on either side. The boxes, instead of it just being a one-to-one comparison, would be whatever sequence position we're currently on plus the 10 nucleotides after it compared to whatever sequence we're currently on or position we're currently on in sequence 2 plus the 10 nucleotides after it. And those 10 nucleotides would have to have at least 8 similar ones in order for a dot to be present. So say this was like A, T, G, blah, blah, blah. So that would be all of the A, T's, G's, everything would be included. Once we move to this next row here, we're not including A in that window size of 10 anymore. So it would be T followed by G all the way up to 10 nucleotides. But since we're still in the first row here, we're still comparing that first 10 nucleotides of that second sequence to determine if a dot goes in there. And it goes so on and so on and so on from that. That can feel a little bit confusing, but we will get into a nicer, neater, cleaner example right now. So here's an example of the differences here. Here we have two sequences that are showing the difference between just comparing nucleotide by nucleotide versus using a sliding window and a threshold. So this first example here is what we get if we have nucleotide by nucleotide. So you can see we definitely have a large area of similarity here, which we can see on that diagonal, but we also get a lot of noise coming up on the off diagonals as well, where we're seeing things that are similar, but maybe not as important to the question that we're asking. So a diagonal line without breaks is always going to show that those two lines are absolutely, or those two sequences are absolutely identical. Oftentimes in dot plots, so if I just do a plot like this, you'll see little regions of diagonals, but they won't necessarily line up all the way down the center. And that just means that you'll have similarity in different parts of your sequences. So you'll see lots of examples of that as well. When we start using a window size, this really cleans it up here. So in this example, we're using a window size of 3. So for this very first box here, what we're comparing is ATG with ATG. And we don't have a threshold defined here, so let's decide it's a threshold of 3, just to be consistent. And that means all the nucleotides in the window have to be the same. So we have an A and an A, a T and a T, a G and a G, which results in a dot. If we moved over to this box here, our window of 3 would be T, G, G, C compared with A, T, G, G. Does A equal T? No. Does T equal G? No. Does G equal C? No. So we don't get a dot there. And you can see that same pattern through all of these different boxes. So if you're ever trying to figure out for any given box, what are we actually comparing? You just need to take that box, so say let's take this one here, above that last dot, go up all the way to the first sequence. That is your first nucleotide of the window, and then you just count 3 nucleotides over since our window size is 3. So these 3 nucleotides are what are being considered for the sequence 1, and we would just go all the way over to sequence 2 here. And again, we'd count 3 nucleotides down to figure out what nucleotides are being considered for that sliding window as well. So in that case, we're comparing TAG with CTA. Again, none of those match position to position, so that's why there is no dot in that window. We have lots of software in bioinformatics that allows us to generate dot plots just by pasting in sequences. So there's one called Dot Matcher that is very, very popular. The website to access that is just right here. And there's one called Dotlet. we are going to use dot matcher to draw a dot plot of two genes representing in the influenza A virus nucleoproteins for the PB2 gene that infects ducks. So we have the two reference sequences there. Down here, it gives us the scoring matrix and the window sizes that it wants us to try. So first, I'm going to open up dot matcher. And then I'll just switch my screen here. Just give me a second. It should look something like this. And I'm just going to zoom in, try to zoom in if it lets me. There we go, a little bit to make it easier to see. So it's a very basic UI. You'll find that often with the bioinformatics tools that our user interfaces or UI kind of remind you a little bit of the 90s. We are not graphic designers, web designers, you name it. We just like having cool algorithms that generate cool results. And if we're putting that on a website, we're often wrapping it in a very, very basic interface. So there's options that we can have for selecting input sequences. You can enter the universal sequence accession number if you have that right here. You can upload a file or you can paste or manually type the data there. What I am going to do in the case of the examples we have is if you just click on the hyperlinks to those sequences, it will take you to the exact sequences we want. So this was the first one here. I'm just going to scroll down because to show you, you might not have ever seen the EBI version of the nucleotide database. So usually we're using NCBI, and equivalently you could search for that accession number in NCBI and you would get the same sequence. But all I'm going to do here is go over to this general side and we can either view the FASTA, download the FASTA, whatever we want. In this case, I'm just going to view it and I'm going to copy this by just selecting the whole thing. I'll right click, copy, and then I'm going to go back to emboss dot matcher and paste that into the first box for the first sequence. I'm going to go over to the second one. Again, I got this just by clicking the hyperlink on the not the PDF. Well, it is a PDF, but the slides. I'm going to go over to view on the right hand side there, click FASTA, and again, I'll copy this, right click, copy, and then paste it into the second sequence. There are keyboard shortcuts you could use for that. So if you're on this page here, you can do Control A to copy it all, Control C, or to select it all, sorry, Control C to copy. And then when you go over to emboss, it's just Control V if you're on a Windows machine. If you're on a Mac machine, it's very similar, but it's just Command instead of Control. So Command A, Command C, Command V. Anything is fine as long as we get this in there at the end of the day. If we wanted to specify our own scoring matrix, we could upload a file here. In this case, if we go back to the slide, the scoring matrix that it wanted us to use was eDNA simple. So I'm just going to copy that from the slides, paste it over here, and it should look like that. You can also type it out. I just don't trust my typing after my first part of this lecture, and I was mumbling my words so much. Down here is where we can change the window size. So the first window size we want to try is 3 with a threshold of 3. So in this case, we just switch that to 3, and we switch the threshold to 3. We shouldn't need to change anything else, but you absolutely can try different settings if you want to. So this output graphic format just means we'll get a PNG out of it. If we wanted to add a title, we could. If we wanted to be informed by email when it was done, we can. Usually you only need that when it's a really, really large sequence. And then we just hit run dot matcher at the bottom and it will run for us and it will create this. Now this looks like chaos. You'll see here that I'm not knowing if any of these things are similar by looking at this. I kind of see a diagonal somewhere in here. And just so you know, it is slightly different orientation than the examples we had been showing before. So we have the one sequence down the x-axis or the y-axis here, and then the one on the x-axis is at the bottom instead of the top, which we show the the sequence on the top is what we had shown in the slides before, but just because it's on the bottom, then we're going to get the diagonals going in a different way. So I can kind of see some coming up. If we saw identical sequences, there would just be a really strong line here, but I'm not really seeing a lot. I'm seeing a lot of noise. I'm seeing a lot of headaches. I'm not convinced here. So if we just go back, we should still have all of our sequences pasted in. And we're just going to change the window size to 9 and the threshold to 8. So that means for every 9 nucleotides that we're comparing, at least 8 of them have to be identical. And again, we're going to run dot matcher, and wow, look at that difference. So this is one of the main tricks, I would say, of bioinformatics. It is so very different than from when we do and run biological experiments. So oftentimes, say you're running a PCR in a lab, your PCR will only produce a result if you did everything correctly. With computers, that isn't the case at all, and it's a really different way of thinking. With computers, generally, they will always produce something for us. And it's up to us to make sure that something makes sense. So with these tuned parameters that we have here, it's a lot easier to see, oh yeah, there is some very strong similarity that we have between these two sequences, as well as regions that might not be perfectly aligned, but are definitely similar popping up as well. And that's because we tuned those parameters down. So looking at that first image, it was like, oh, I can maybe see something if I wanted to, but I'm not super confident. Tuning those parameters, making sure that we're looking at a clean and nice image, we get a result that we could be a lot more confident that those two sequences are similar. So it's up to us as the bioinformatician to ensure that the output that we're seeing makes sense given what we know about the algorithm we're using and the biological question at hand. Since we knew those two sequences were coming from a similar gene or similar protein, we could have a fairly high confidence that they were supposed to show some similarity between them. So if we go back here to the slides, remember it was 2 genes representing the PB2 gene. So we know that they're likely going to have some sort of similarity with them. So when we saw that first image and it was just all chaos, that was a good indication that maybe we needed to go in and tune those parameters. Similarly, sometimes we can put things in and we can get results and they don't make biological sense. And we need to be able to put on that biology hat to say, hey, even though the computer spit something out here, it doesn't mean it's fact and it doesn't mean it's right. And our intuition about the biology happening behind this suggests we need to go in a different route. And that's a really important job for bioinformaticians to do. So this is just a screenshot showing what we already went over of how you would do that, just for record purposes. Moving along from dot plots, we're going to start talking about sequence alignments, which is why we are here today. So as I mentioned before, I'm fairly confident in saying that sequence alignments are the most common operation in bioinformatics. They were certainly the most common operation I did when I was a bioinformatician, and they allow us to identify which parts of the sequences are similar. So unlike a dot plot where it's just a graphical visualization of what's similar, this will give us the actual nucleotides that are similar. So here is an example. If we have a gene fragment called TAG, I don't need to read that all out to you. I don't know why I am. Assume we know that the following occurred throughout the evolution. There was a mutation change from the 4th nucleotide to a T, and the second last nucleotide it mutates to a C, and then there's an insertion after AT. Since we need this for the next slide, we'll just do this together instead of me having you do this on the discussion board. So what I'm going to do is just type out that sequence as it currently is. So TAG, CC, GC, AC, GTA, TTAAC. And then I'm going to apply these mutations that we know that occurred based on what happened here. So 4th nucleotide, so 1, 2, 3, 4, which is this C here, changes to a T. The second last nucleotide, which would be this A here, mutates to a C. And there's an insertion after the 6th nucleotide. So 1, 2, 3, 4, 5, 6 of an AT. So that should be the sequence that we get from all of those mutations that occurred. I am just going to copy this. Oh, I'm so used to using Mac compared to my Windows machine, but that's okay. And we'll use this on the next slide. So here shows the sequence that was mutated, and we can visualize the evolution in the following way. So we have both sequences lined up here. Anything that's different is highlighted with red, which might not be the easiest to see, so I apologize. And then you'll see this big insertion on gene 2 has these dashes on gene 1. And what we call that is a gap on the other gene. So whenever we have an insertion or a deletion, one of the other genes in the alignment will have what we call a gap, which we represent with dashes, just meaning that those nucleotides don't exist in that sequence. So effectively, whenever we're doing sequence alignment, what we want to try to do is line up the sequences together as best as possible to minimize the number of mismatches and then also the number of gaps. Given that we have this, I want you to think about what kind of clues in the alignment could we use to determine similarity. I don't think I have this one. I'm just checking on the discussion board off to the side here. I don't think I have this one written down there. So think about them and then we'll flip to the next slide, which will give us the answers. So the more nucleotides that line up, the more that the sequences will be similar to each other. So the more times we see things like this, where CAC GT and CAC GT are exactly identical, that's a great hint that the sequences are similar. The less mutations that we see, so things like this here, these single nucleotide variations, the more similar two genes or two sequences will be. If there's lots of mutations, then it's less similar. So it's just those two ways of thinking about it. And then the final thing is the more gaps like this that are present, the less similar those two sequences are. So gaps indicated that there was some larger insertion or deletion event, which doesn't necessarily mean they're not similar. It's just that they're less similar than two sequences that don't have those gaps involved with them. So typically we don't have information on the mutation events that occurred throughout evolution like we did in that example, and we're often using sequence alignment to try to find out that information for the ultimate goal of identifying whether or not two sequences are homologs. The one big piece of caution here that's related back to what I said about dot plots is that alignment and similarity can be determined for non-homologous sequences as well. So if we throw any two sequences into these algorithms, we will get an output. The computer isn't going to tell us, hey, by the way, you put in some sequences here that really don't make sense to be potential homologs, so I'm not going to give you an answer. No, it's going to say, oh, I got a bunch of text and a bunch of text, and I know what to do with all this text, and here's your result. And so it's us, the bioinformaticians, that have to say, does this make sense that these two sequences would be homologs? Is this even a reasonable question for us to be asking? And that's when we tap into our biological knowledge and really try to ensure that we're asking the right questions of whatever problem we're trying to solve. So that biological hat is so important. And you'll hear me say that over and over again in this class. There are some unusual situations. So we have, or sorry, not unusual situations. The usual situation that bioinformaticians are faced with is that we have some sequences and we want to determine the best alignment to determine the similarity and predict the most likely positions where mutations occurred. Because again, we don't know the events that occurred evolutionary. So we're just predicting based on what we see in the alignment. But a really important question is, how do we determine what's a better sequence alignment? What's the best possible sequence and alignment? We need to have some sort of mechanism in order to do that. For instance, there's lots of different alignments that we could get between these two very long sequences, and I'll resist my urge from reading them out loud. I feel like I'm already doing that too much and it's a bit cringy. So here are some examples. We have the sequence 1 at the top, sequence 2 at the bottom. We could have just a really big gap here. I think there's a couple nucleotide variations that we could see in that. The gaps could be in different spots. So again, we have some things that are lined up at the beginning, a couple SNPs, some gaps, some more alignment with a few single nucleotide variations, right here, followed by more gaps. We have this one here, which has lots of little gaps interspersed with some alignment and some variations. And then we have this one down here, which is all gaps and no alignment between the two of them. And when we look at all of those, which one looks like the best alignment and why? What do you think? Before I move on to the next slide, I want you to get an idea of your head of like, what one looks like that? If I had to pick between any of these, which ones would I pick? I'm probably not picking this one. I'm probably not picking this one. Maybe one of these ones. I have a feeling of what I would pick, but I also have been a bioinformatician for a long time, so I feel like I'm biased, and I want y'all to make your own decision there. So what we often do to help us remove our own internal biases of what the best alignment is we use something called an alignment score. This is just a number that represents the number of mismatches and the number of gaps that we have in an alignment. It's a bit of the it's the inverse idea to an edit distance, where we were talking about the minimum number of operations needed to convert one sequence into another sequence. Effectively, we go through line by line or column by column of that alignment, and we assign a score for each position of that sequence in the alignment, and then we sum that up for the entirety of the alignment. And we'll get into some examples here. So I'll go back here and I'll just say what I mean. So if we effectively treated each position or each nucleotide or gap in this alignment as a column, for this T and T, we would have some number that we gave to a match, and we would go through and we would figure out, do these two nucleotides match, mismatch, or are they matched with a gap for every single position in that sequence, and then add them up at the end. So oftentimes what we use to figure out what those numbers should be for things like a match, a mismatch, and a gap is something that we would call a DNA scoring function. We use different scoring functions for DNA and for protein because proteins are a little more complicated, have a little more intrinsic preferences of what kind of amino acids they'd be subbing with other amino acids, but we'll get more into that later. For DNA, it's a lot more simple. We have both simple DNA scoring functions where there's just a straight penalty for a gap. And then we also have more complex DNA scoring functions where they have a gap opening penalty and a gap extension penalty. And we'll get into more on that later. So what would the score for this one be, given that we have that scoring function here, where a match is a +1, a mismatch is a -1, and a gap is a -2. So we're going to go through here. I'll try to line it up as best I can. Let me just move this a little lower. First, we have a T and a T, so that's a 1, followed by an A and an A, so that's also a 1, followed by a G and a G, so that's a 1. Then we have a C and a T, so that's a -1. A C and a C is a match, so that's a 1. Let me just get rid of that. A G and a G is a match, so that's also a 1. And then we have a whole bunch here lined up with gaps. So we have 1, 2, 3, 4, 5, 6 gaps. So that's 6 minus 2. So 1, 2, 3, 4, 5, 6. It's hard for me to do all this mental math this late at night, but we'll get there. After that, we have a match, 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 all the way up to this A. So that's 1, 2, 3, 4, 5, 6, 7, 8, 9. Plus ones. 1, 2, 3, 4, 5, 6, 7, 8, 9 plus ones, followed by a mismatch, which is a -1, and then a match, which is a +1. Now we have to do the mental math of calculating all of this together. What I will do, just because I have this copied or typed out instead of on a whiteboard, and I usually, if we were in class, would just ask y'all to tell me what it was and do it on your phone while I talked about or blabbered on about something else. But I don't feel like it's the most wonderful to watch me run this through a calculator, but that's what I would recommend doing if I was doing this for an assignment. What I'm going to do is just remove things as they cancel each other out. So we have a -1 here and a +1. So we'll get rid of that because those two will equal 0. We have a -2 and 2 +1s, that will equal 0. A -2 and 2 +1s, that will equal 0. A -2 and 2 +1s also will equal 0. This will equal 0. This will equal 0. This will equal 0. And this will equal 0, resulting in an answer of 1. I think that might have been easier actually to watch me just type it through on a calculator. But that should be the answer if my mental math was correct. If it wasn't correct, of course, correct me in the discussion boards. Now what I want you to do, and I have this posted on the discussion boards as a prompt, but go through the other alignments, figure out the scores for those, given that same DNA scoring function that we had on the previous slide. And from that, can we determine what the best alignment is? Does that match with your intuition from the previous question? So when we're trying to find a good alignment, we're going to have a few things we want to keep in mind that we've talked about. The more that nucleotides that match, the better. The more nucleotides that mismatch, that's worse. We don't want that. The more gaps there are, that's worse. We also don't want that when we're looking for similarity. So remember, good and bad here is in the context of, are these two things similar? So our typical scheme is going to be a positive match score for the matches, and then negative mismatch score, and an even more negative gap penalty. There's been lots of research into what makes good DNA scoring functions for different levels of relatedness, and so we'll probably give you a lot of that information, but that's kind of some of the intuition that we're going to see here. The general assumption is that a good alignment is one that's going to reflect actual homology if the sequences are homologous. So that's a big if, and again requires our biology hat to help confirm. So why do we need computers to help us find the best sequences? Well, this actually, in theory, we don't, but I'm sure you've already watched me make mistakes as I've gone through this lecture when it comes to aligning things or even those edit distance. Computers are a lot better at doing repetitive tasks and not making mistakes than humans, especially when they're dealing with lots and lots of possibilities. So the number of possible alignments grows exponentially in terms of the length of the two sequences. So for two sequences with 11 nucleotides, there are over 700,000 possible alignments. Yes, a human could do that. I'm not signing up for that job. I wouldn't recommend anyone else sign up for that job. I've always joked that I'm trying to automate myself out of a job. I haven't done it yet, but anything I can get a computer to do so that I can have more time to do other things, I am game. And sequence alignments are one of those things. So there's two main algorithms that we use for sequence alignments typically in bioinformatics. Remember, we had our dot matcher for generating dot plots, but that's just a visual overview. These algorithms for sequence alignments will actually show us what portions of the actual nucleotides, like the A's, the T's, the C's, and the G's, that actually align or have gaps. So the Needleman-Wunsch algorithm is one of them, and the Smith-Waterman is the other one. We're going to use Needleman-Wunsch to perform an alignment on the myoglobin sequences from human, and we have the two accession numbers there, and then the scoring penalty or scoring function down there. So if I just click this, and then again I just need to go over and swap this to this, and you'll see another very beautiful user interaction field where we have all of these lovely boxes, very similar to the dot matcher one that we had before. We need to go in and we need to just grab some of these other sequences. You might be tempted to just throw those accession numbers in here. Oops, that's not the accession numbers. That was my scoring functionality or work from last time. And I'll show you what happens when we do that. So if we just put those accession numbers from the slide in, do we actually get what we want? And for our scoring function, we wanted a gap opening penalty of 5, a gap extension penalty of 5, and then we don't need to add anything here. We don't need to add anything here. And then we just need to figure out, what do we actually want here for our matches and our mismatches. So it told us to use a match and a mismatch of 5 and 4. So we can go in and we can double check, like, what does the eDNA full, if we just Google this, eDNA full matrix look like? And it should bring it up for us and show it. I think this is the right one here. Yes. So we have all of our nucleotides up here, and then some of our different symbols that we don't have to worry about. And so we can see here that our matches, which would be down this diagonal, are 5s, and our mismatches are -4, which is exactly what we wanted. So we can go back to our Needleman Wench, and we just have to type in here eDNA full. Oh, and my cap locks is off. eDNA. Wow, and my typing. eDNA full. There we go. And if we try to run Needle with this, you'll see we get errors where it says, we don't know what the heck you were trying to open. That just means that the sequence accession number that you added here is not a universal sequence accession number. So instead, we should go over to NCBI, nucleotide, go into that website, search for those accession numbers, and grab our FASTA. So here's our first one, Homo sapiens myoglobin. Click FASTA up in the top left corner, and we can just copy that FASTA. And go back over to Needle, paste it down there, and get rid of that accession number. And the same thing for the second one. Paste that in. Clicking FASTA over in that top left corner. Copying and pasting, and then just deleting that accession number. Again, we already have our gap opening and gap extension penalties that we wanted, and we have the match and mismatch values we wanted by using that eDNA full matrix there. So we just click Run Needle. Now we shouldn't see any errors this time, and we don't. So up at the top here, it just gives you an indication of what parameters were used to run Needle that you specified. So you'll see we have our gap open, our gap extend, our eDNA full, our sequences that we put in. And then as we go down, we get more information. So we have 2 aligned sequences. We have an overall length of 1,170 base pairs. We have the percent identity, the percent similarity, the percentage of gaps, and then our overall sequence score. And as we scroll down, this is where we see what's actually aligning and what isn't. So it goes in increments of 50 base pairs. We'll have one sequence on the top line, one sequence on the bottom line, and then you can see what was in those individual sequences, what was a gap, what was a match. So you'll see that matches are indicated there, and then the mismatches are indicated with a dot. And we can go through the whole sequence and see exactly what they did. You saw how long it took me to do those 11 nucleotides. This was done before I had gotten to like the plus or the minus 2s in the 11 nucleotides, right? Much, much better, in my opinion, than watching me go through and do this by hand. I'm much more confident in the abilities for this to run successfully. So that should be everything you need to know about running Needle. So pretty straightforward, at least in my opinion. But again, I've done that a lot. It's okay to have questions about it. It's okay to ask your lab instructor about it or me about it. We're here to help. So the question is, why would we want to potentially use different values for opening a gap and extending a gap? So far, we've seen only examples where the gap has the same value. So in the previous example, by saying the gap opening penalty was 5 and the gap extension penalty was 5, that just means any gap has a penalty of 5. So would be a -5 when calculating that score. So why would we potentially want to use different values? And think about this in terms of your biological hat. When in biology, does it make sense if we had two sequences that we would expect there to be a lot of gaps in one sequence versus the other? What if we were comparing the gene sequence to the mRNA sequence? Would there be situations in there where we'd expect large chunks of gaps? Probably, with our introns and our exons. So remember, our mRNA sequence would have our exon or our introns spliced out and only have exons. So we would expect a whole bunch of large gaps. So while we wouldn't want to just be throwing gaps in wherever, we wouldn't want to have that gap penalty really, really low. We would say once a gap has started, to extend it is something that we would expect to happen. And similarly for a lot of our insertions and deletions that happen in our gene substitutions, once that gap opening has started, it's likely that there'll be a chunk of nucleotides that are either inserted or missing that we need to account for. So the simple gap penalty, where we're just doing one value for all gaps, could generate sequences or sequence alignments that are similar to this up here, where any time that we just are mismatching a nucleotide, we just put a gap in instead. If we don't have a lot of distinction between our nucleotide score and our gap score, that's really likely to happen. And again, remember, the computer is not an expert in biology. We are the experts in biology. So we need to be feeding it and tuning parameters so that it's doing something that is reasonable. Instead, if we had a gap opening that was really high, but then once it started, it was less penalized, you see more gaps that are longer, but can like in one part of the alignment, which is usually more realistic for biological use cases. So these two alignments have the same score, but when I'm looking at them, this to me seems a lot more likely, the second one down here, for a biological use case than this one up here. This one up here looks like a hot mess. Like, that's a lot of errors to be thrown in a very small little area that were caught by some of the correction mechanisms within cells. So I am highly suspicious of that alignment. Whereas this one here, okay, maybe there was just a really big insertion that happened, or a deletion that happened. That makes sense. We've seen that before. I can get on board with that kind of alignment. So these more complicated gap opening and gap extension penalties are actually something called affine gap penalties. And that just means that the gap opening and the gap extension have different values. And again, this makes it more likely that we'll get adjacent gaps in a sequence alignment rather than multiple non-adjacent ones just throughout. Usually representing insertions or deletions or things like introns that have been spliced out, things like that. So consider this following alignment that we have here. We have it down here. There's 3 different sequences that we are working with. We have some gap penalties and we have some gaps that are bigger in both the second and the third sequence alignment. So with that, if we assign a smaller gap extension penalty than an opening penalty, we're going to get lower sequence alignment scores when we have those continuous gaps within our sequence alignment. So this here walks you through for a match score of 3, a mismatch score of 0, a gap opening penalty of -2, and a extension penalty of -1, we'd get a score of 9. I want you to go through these examples here and kind of work through that exact same situation where we have this scoring matrix up at the top and then calculate the scores of these 2 alignments here. I feel like you've seen me walk through this before, and the only difference is going to be when we have a gap opening is going to be -2. So anytime a new gap starts, we'll have -2. And anytime a gap is extended, so like we'd see down here, this first gap here would be -2, but then all of these other gaps would be -1. Whereas all of these individual gaps, since they're off by themselves in the lonesome, would all have values of -2 associated with them. So for both of those, you can get your scores for those, and then you can see, does one of these actually have a better alignment when we use those more fancy affine scoring penalties. In general, there are two types of sequence alignments. We have global sequence alignments, which align across the entire length of the sequences, so from beginning to end. This is what Needleman-Wunsch does, so it goes from the very first nucleotide to the very last nucleotide. There's also local sequence alignments, which we'll get more into next week. And these are just alignments across sub-parts of the sequence. So it just looks for things that are very similar in portions of the sequence. It doesn't necessarily take the whole sequence into consideration. And so that algorithm is called Smith-Waterman. It's very useful when homology only exists in part of the sequence. So this happens a lot with like protein domains. Y'all know from your biology courses, there's lots of different kinds of protein domains. We can see those in different types of proteins. So a gene might code for a specific kind of protein domain that we already know the function of. We would use a local alignment to identify that protein domain in, say, an unknown gene, help give us some insights into the functionality of that. So this gives you kind of an overview of what we can see with some of those protein domains. We have a couple of different genes here and their protein domains called out into those little bubbles. So you see we have things like SH3 domains, SH2 domains, and they're kind of all throughout, interspersed between all of these different sequences. If we use Needleman-Wunsch to try to align them, it wouldn't necessarily identify that, hey, we have an SH3 domain here and here in this sequence and here in this sequence. It would try to go from the beginning and align this up with this beginning bit and in this middle bit, and then there's probably some big gap somewhere over here. Needle would really, really struggle with trying to identify the similarity of the SH3 region there. But if we used Smith-Waterman, we would actually be able to pull out that, hey, it looks like there is an SH3 domain in that top sequence and in this bottom sequence, giving us a hint into what kind of protein domain we might have going on there. And same examples are kind of shown on all of these other ones as well. So for our final exercise before we get into substitution matrix, we're going to use Smith-Waterman and Needleman-Wunsch to perform global and local alignments of these two accession numbers. So if I click open into those, so let me get rid of some of these. And again, we are going to have a very, very similar overview as to what we've seen previously with our beautiful UI. Y'all will come to love these 90s UIs, I promise you. All we're going to do, again, is grab those sequences from NCBI. So I'm going to copy the first one, head over to NCBI from when we were using it previously, paste that in, click FASTA, and grab this sequence. Control-C, I'll paste it down here. And then again, I'm just going to grab the other one. So the other one is also a protein. Over here. Search. FASTA. And here we go. And then back over to Needle. Let me just double check. We're just going to use all the default scoring functions. So we'll just let it use the default. We don't need to worry about specifying it here. We don't need to worry about specifying gap opening or extension penalties. And we're just going to hit run Needle. Again, went way quicker than if I was doing this by myself. So it tells us the length, the identity, the similarity, the gaps, and the score. And you'll see here that the identity is slightly different than the similarity because we're using a protein sequence. So identity and similarity with protein sequences will be different than when we're looking at nucleotide sequences. They will be the same. That is because they take into account the types of amino acids that are matching. So it's slightly different. We will get into it a little bit more in the next video. So here is what our alignment looks like for needle. We have, you'll see it starts from the beginning, a very, very large gap. Then it's identified some area that looks slightly similar. So there's some that are lined up, lots of mismatches that are happening in there. And then again, it goes all the way to the end. Since needle is a global tool, it's very expected that that would be the output. Now instead, if I just go back here just to grab these sequences, if we run this in a local tool, I'm just going to go Control A, Control C, paste that over. And then again with the second one, Control A, Control C, paste that over and run this. And I'll rerun needle as well so we can compare. With water, we're just doing a local output. So you'll see we have a much higher identity and similarity, much lower number of gaps, and it's way shorter than what we saw with needle. So you'll see that the top sequence starts at the base pair, the amino acid 109 and ends at the amino acid 221, whereas the bottom sequence starts at amino acid 4 and ends at amino acid 117. So again, this is pulling out most likely a region of similarity related to that protein's function. Whereas when we look at this sequence, it's like, ooh, I don't really know what's going on. That kind of starts around the same, but this here looks a lot cleaner and a lot easier to understand. It's likely that our sequence 1 is a protein that has another subdomain in it that isn't represented in sequence 2. So when we run that through needle, it's like, these don't really align at all. What are you trying to do here, you bioinformatician? This is weird. And whereas when we run this through here, it's pulling out that one region that is similar, which is likely a protein subdomain region that comes out. So oftentimes when you're working with proteins, you're going to want to be using things like water instead of needle for alignments. Oftentimes when you're looking at things like, saying, trying to align a gene to a whole genome, you would also want to use water because otherwise it's going to have so many gaps in it if you use needle. If you're trying to just you have a way shorter sequence compared to a very, very long sequence, often going to want to use water instead of needle. So just to keep those types of things in mind. I need a quick break before we get into the very last section on substitution matrices. So we'll chunk that out into part 3 of the video. We'll see you there.